So Science Corps is a nonprofit that gives young PhD scientists the opportunity to do science outreach and educational outreach abroad. And something that many of the students of those fellows are interested in is how and why someone becomes a scientist, and actually also issues and the ethics around gene editing. So I am very lucky to have the opportunity to be speaking with Professor Jennifer Doudna, who's an esteemed scientist at UC Berkeley, and is taking the time to answer a few questions on these topics.
Sounds good.
So I want to start with when you were a child. Do you remember anything about what led you to be a scientist?
I liked puzzles. I liked math. I enjoyed my chemistry class in high school, and I decided that I wanted to understand the chemistry of living systems. I wanted to understand how cells become cancerous. When I thought about a problem like that when I was a kid, I always wondered, well, why? What molecules are responsible for that change? And that's sort of the type of science I always wanted to do.
Hmm. And what do you like most about being a scientist?
Oh, being a scientist is a fantastic job because every single day I get up and I think about questions that I'd love to answer, and I get to come to work and work with some of the very best, smartest young people to figure out how to answer them. So every day it's solving a series of puzzles.
Yeah. And what would you tell a young person, a young student who is considering becoming a scientist about either how they might do that or what they might expect?
I think it's like anything else in life. I think you have to decide you want to do it, and then don't let anybody dissuade you from doing it. So and I can think of many times in my career when I had doubts about myself or my career choice, or people questioned my career choice. And at all of those times, I somehow worked through it. I knew in my heart that it's what I really, really wanted to do, and I just kept doing it.
And could you tell us about a time that you failed, not just a challenge or a setback, but a real failure, and either as a student or a scientist, and what you did to keep yourself motivated and to make it through that?
Well, I can remember numerous times when things didn't work in the laboratory. That sort of goes hand in hand with doing research. You have an idea, a hypothesis about something, and you go to do the experiment, and it doesn't work. And the question is always, well, why not? Was my hypothesis wrong, or was my technique somehow flawed, or was there some technical error? Did I make a mistake? And I think at those times I've always resorted to a few different strategies to keep myself going. One is reminding myself, why am I doing this? Stepping back and saying, what's the bigger purpose here, and what is the importance of this experiment? And sometimes it makes you say, well, maybe that experiment actually isn't so important. There's something else I should be doing. Or sometimes you say, no, I really, really, really have to get that experiment to work, and so I'm going to figure out a different way to do it. And when I was a student, the way I managed it is, I love gardening. So I always had even when I was in rental apartments moving around Cambridge, Massachusetts, where I was a student, I always asked my landlord, could I have a little garden in the yard? And that was a huge help to me, because many times I would come home from the lab frustrated, and I'd go out in my garden and I'd do something there, and it would just give me the kind of the instant gratification, satisfaction of working on something in nature, and then I'd have the strength to get up the next day and go back to the lab and try again.
That's really cool to know. And so on the side of gene editing, why do you think some people find gene editing and CRISPR-Cas a controversial topic?
Actually, I think the topic isn't controversial. What I would say is that certain uses of gene editing are controversial, and in particular, it's something that's been in the news a lot lately. Is, well, two things. One is using CRISPR-Cas gene editing to modify the DNA of plants, and countries sort of at the level of regulatory agencies arguing over how we regulate plants that are produced in that fashion? Are they called GMO or not? And what does that even mean? So that's one area. And then the other is in the area of human germline editing, meaning making changes to DNA in eggs or sperm or embryos that become heritable. So that means they can be passed on to future generations. So that's a really profound use of gene editing. If you think about it, it means really altering the whole human gene pool and the future of human evolution, if we took it to that level. But I think at the stage we're at right now, it's more of a question of, is there any medical use of that kind of gene editing? And if so, how does one approach it in an ethical fashion?
Right.
And there was a recent announcement out of China with a scientist proceeding into clinical use of human genome editing that I think has made many people say, well, that's not right. That's clearly an irresponsible use of the technology. How do we take a step back and ensure in the future that scientists respect ethical boundaries? And that's what's happening right now. So I think the silver lining in that case maybe is that there is a real international effort and an urgency to having that conversation and putting in place very, very clear guidelines.
Right. So yeah, I think you answered that question and the next one too. I think just a final question I want to ask you is about science outreach in the developing world, increasing science capacity, and whether you think that's an important issue, and if so, why?
I do. I think it's very important, and I really salute you for your efforts to help with that, because I think that in my experience, the more diversity we have in science, the better it is. Right? People come from all around the world. They come to my university, our university, UC Berkeley, right? A public university. We have students from every part of the world that come here because they want to learn, they want to study, and they want to do great work. And I think that spreading that the idea of science and how we approach scientific questions, inviting people from all over to come to that endeavor with their unique perspective and their unique set of questions and probably different ways of answering those questions is only to make science better in the future. So the more we can do of that, I think it benefits humanity.
Great. Well, thank you so much for taking the time to speak to me, but also the students in India and the Philippines that will be hearing this. And so, yeah, great. Thank you so much.
Fabulous. It's a pleasure.